Okay, so in this lecture, what we're going to talk about is something that has probably been on your mind, and that is where, how do our cells, the B cells and T cells, um, end up being able to recognize their antigens and also being able to recognize MHC1 and 2. I mean, how does that done? I mean, because when you think about it, what if you you might have probably realized that what you see, what the immune system has is a pre-ordered capacity to recognize uh, pathogens and things that are foreign when they come into when they're come into contact with that system. So and be able to adapt to them. So there's a pre-order, there's a pre-capability that you that the system has, and then there is ability to change that and adapt to the system too. So where does that come from? So the sort of two take-home messages from this section um, are going to be one, B cells and T cells that react with self-antigens have to be eliminated. So part of that is the fact that we need to actually eliminate B cells and T cells that might react with us. The second part is that we won't cover in this lecture, we'll cover in the next lecture, will be effective immune response depends on organs that bring antigens, antigen processing cells, and the T cells together. So those are the two big concepts from this section. So if we work at the primary lymphoid organs, so there are primary lymphoid organs and there are secondary organs. These fill two roles. The first is the differentiation of B progenitor cells and lymphocytes into B cells and T cells, fully functioning B cells and T cells. They also serve to um, determine the ability of B cells and T cells to recognize the foreign antigens. So they set sort of the universe of what is potentially foreign. Um, and that universe is defined primarily by those antigens that are referred to as self and eliminating the ability to recognize self antigens. So if we look at the primary lymphoid organs, what we would find is that those organs are uh, here in red, and these would be the bone marrow, which would be here. And so the bone marrow, and we've already discussed the bone marrow is where stem cells, the bone marrow stem cells are, and the progenitors all come from, and the myeloid end, these are where those cells come from, and the lymphoids, where B cells and the thymocytes come from. And then the other organ is this little organ up here called the thymus. And the thymus is the other part of that system, and this is where T cells are going to mature. So these are referred to as the primary organs. So if we look at the bone marrow, let's talk about B cells first. We have progenitor cells are then interact with stromal cells, and these are nurse cells as we talked about before, I mentioned before. These are stromal cells are basically provide the microenvironment, they release cytokines and provide ways, nutrients and things that allow factors that allow to maintain those progenitor cells in their state so that they don't die in that place. We're able to move on to the next step. And in that, those progenitor cells will proliferate into pre-B cells. And so these are cells that are not quite B cells yet, but they're into, you'll see proliferation into several of these cells, proliferation to a number of these cells. And each one of these pre-B cells is going to have a particular display of an antibody. It's going to be able to display on its surface antibody that will recognize a potential antigen. Of those, we're not really sure how this is done in a lot of detail, but roughly 75% of the pre-B cells are eliminated because they don't express antibody very well, or the antibody they do express seems to react with self-antigens, and so therefore those pre-B cells are eliminated. So there's a lot of cell death that actually happens in which you eliminate these cells from that system. Uh, the remaining cells, and this all happens in the bone marrow, the remaining cells then leave the bone marrow into the central sinus and develop from immature to mature B cells, from which in which those cells now are able to have been produced and are able to potentially recognize a foreign antigen. And from there, they then migrate into the bloodstream, and then will distribute to the body. Now, the other organ is thymus, which I mentioned before. It's up here. And so the thymus is going to be governed the interaction. There's two critically important issues when we talk about this, and this concept is referred to as tolerance. And when we talked about when I just mentioned the B cells, they're becoming tolerant. Essentially, the B cells are being tolerized to you. In other words, you're eliminating cells that are self-reactive and will react to potential antigens that may be on the surface, or inactive antibodies on the surface. In the case of T cells, remember, it's the interaction between MHC1 or 2 and the T cell receptors. And in that context, what you want is tolerance. In other words, the cells have to be tolerant. The concept is how do you develop tolerance to your system, because you contain a tremendous amount of antigens. There's MHC1s that are expressed on the surface of, for example, in the surface of all nucleated cells, but they contain self-antigens. All right, so that shows that they don't have anything foreign in them. How do you become tolerant to that? How do cells just don't interact with those things, and don't end up killing our own cells? And so there's two concepts. The first is positive selection, and this refers to the selection of T cell receptors, and therefore T cells, that bind to MHC1 and MHC2. So these are T cell receptors that will bind there. And let's say this, and that requires, if we see this as a simple, as just a simple, set of interactions between MHC1 and MHC2. And remember, they interact on those outer loops, right? The loops that are outer, and then the edges of the MHC2. And so what you're interacting with is how T cell receptors recognizing and interacting with MHC2s and MHC1s in those areas, right? And so the areas where they touch each other, okay? And if you go back to the structures where we looked at previously, remember the loops. There are loops on the outer area that interact with those alpha helices on the MHC1 and MHC2, and there are loops and those MHC1 and MHC2 alpha helices interact with the T cell receptor loops. And so these interactions need to be selected for, positively selected for. The second concept is referred to as negative selection, and that's a selection in which you have the MHC1 and MHC2, in which the MHC1 and MHC2 has a self-peptide that's displayed in that is bound into that MHC1 and MHC2. And so in this case, you want to eliminate T cell receptors that bind to MHC1 and MHC2 that have self-peptide. So you don't want them to bind there, right? You don't want them to bind to this T cell receptor. They may be able to bind here, but with the peptide, you don't want them. They don't want them to bind. And so any T cell receptors that do bind, okay, so the ones that do bind in this area, would bind to those T cell receptors, you want to eliminate. Let me repeat that. You want to eliminate MHC1, MHC2 self-peptides complexes no, sorry, you want to eliminate T cell receptors that will bind to MHC1, MHC2 self-peptide complexes. You want to eliminate T cell receptors and those T cells that would bind to MHC1, MHC2 self-peptides. That makes sense because you don't want them recognizing and going off and initiating an immune response and start killing your tissues. So that's referred to as negative selection. And so if we take how this happens, then remember the thymocytes are going to leave the bone marrow, and they arrive through a vessel into the through the bloodstream, through the thymus, and the thymus is composed is sort of two broad units. One is the cortex and one is the medulla in the thymus, and there's blood going in and blood going out of the thymus, and these thymocytes will then home in and enter the cortex. When they enter the cortex of the thymus, they will be encouraged, and they will undergo they'll undergo T cell receptor expression and diversification. In other words, they'll start expressing their T cell receptors. So the thymocytes all kind of look similar when they get into the cortex. When they get into the cortex, they start expressing their T cell receptors, or not expressing them, or expressing them very badly, or the T cell receptors are not functional, but they'll express whatever they're supposed to express. They do that, and there'll be productive rearrangements. Remember, this process is there's essentially, when you go to the thymocyte into the T cell with the properly folded T cell receptor, what happens is there's a process of rearrangements, of recombinations to produce a whole plethora of different T cell receptors, and therefore, each T cell receptor defines a particular T cell. So the T cell specificity is defined by the T cell receptor, and they'll undergo a large diversification and production of different ones, of different T cell receptors, and therefore different T cells. Those will then also be encouraged to express, they will express CD4 and CD8 simultaneously. Now this is important because the T cell receptor that's expressed on the surface, it doesn't know whether it can bind to MHC1 or MHC2 yet, right? And so therefore, at this stage, the pre-T cell will express both CD4 and CD8 on the surface, because it needs those. If it binds to MHC1, it needs CD8 on its surface. If it binds to MHC2, it needs CD4 on its surface. And so under these conditions, you get positive selection, in which there is expression in the endothelial cells. Let's see if I have it here. On the endothelial cells of MHC1 and MHC2. These are actually these are cells that are the only other cells in the body that will express MHC2 are in the thymus. They're not for antigen processing, but they express them. They express them on their surface, and the T cells will interact with them. Interact with the MHC1 and MHC2 that's on the surface, and those that don't bind very well, or don't bind very tightly enough within whatever that restricted window is, will undergo apoptosis and be eliminated. So apoptosis meaning programmed cell death. And so if they're not selected, if they don't bind tight enough, if they don't bind to those MHC1 or MHC2, then they will undergo programmed cell death and be eliminated. And macrophages and not will destroy them. Now, now that they express this MHC1 and MHC2, and there's the selection by these endothelial stromal cells that are in the thymus, then these cells will now undergo an interaction with them in which, if they bind too tightly to the MHC self-peptide on dendritic cells, then they will be eliminated. And so in the system where you have these T cell receptors, they're going to interact with dendritic cells that have MHC 1 and 2 on their and self-peptide. They are going to be eliminated. So if they bind too tightly to those, that's referred to as negative selection, and you get loss of those cells and destruction of those cells. Now, so just to make sure you know, so it's the MHC1, MHC2 selection by these thymic stromal cells or epithelial cells that you see for positive selection, and then for negative selection, it's binding to this MHC1 self-peptide dendritic cells. And so as you can see, you've got positive selection for MHC1, MHC2 binding, and negative selection, elimination of ones that bind to self-peptides that are displayed on the surface of the dendritic cells. So once that occurs, and a cell basically has the ability to bind to MHC1, MHC2, but not self-peptide, it makes it through this gate. There's no binding to that cell, then it then will progress out into the medulla, and then what happens is it will essentially become what is called MHC1 restricted or self-tolerant, in which the ones that are able to bind to MHC1 the tightest will only start expressing, will express CD8. The CD4 expression will be eliminated. Those that binds to MHC2 will then only express CD4, and so therefore they'll eliminate any CD8 that they're expressed. And so now you have mature T cells. These are naive, right? They haven't interacted with any antigen processing cells, and they're referred to as self-tolerant because they should not interact with self-peptides displayed on MHC1 or MHC2. Now there are other, this is sort of a first pass, and sometimes these do happen. Sometimes things do happen in which you still see this, and we'll come to that in a second, how that is adjusted or dealt with. And these will then go to the enter the bloodstream, blood vessels, and start distributing to the secondary organs. So just to recap, you have positive selection for MHC1, MHC2 binding, negative selection for MHC1, MHC2 self-peptide binding, then elimination of CD8 and CD4 expression depending on whether they bind to MHC1 or MHC2, and then migration from the medulla into the blood vessels. So what happens if a T cell, if it kind of makes it out and it is able to bind, for example, to an MHC2 peptide or MHC1 peptide? Well, it turns out there's another way of controlling or reducing the inadvertent or the unwanted interactions between a T cell and self-peptides. This is also a way that the immune system restricts the actual activation of the immune system, because one of the issues with an immune reaction is that it can get out of control, and so there needs to be sort of stops on it, in which if you do have an unregulated activated immune response, you could literally die from that. Essentially, because of the inflammatory response would get out of control. And so there's sort of two things. One is dealing with Potential for T cells to interact with, again, MHC1, MHC2 self-peptides, um, uh, and to controlling that immune response so that it can go so far but not get out of control. And that turns out to be, that suppression turns out to be um, primarily maintained by another set of T cells. These T cells are referred to as T regulatory cells. And they are, interesting enough, they're CD4 positive. They express IL-2 receptor on their surface. And they have this transcription factor, which is the most important thing for identifying them. It's called FOXP3. And it's a transcription factor that's characteristic of T regulatory cells. And so you don't find this. So if I have a bunch of T cells and I want to know how much the percentage of T regulatory cells is in those, I would look for the expression of FOXP3, because neither CD4s or what we consider our helper cells, Th cells, or cytotoxic T lymphocytes express this transcription factor. Now, what this transcription factor does is it produces transforming growth factor beta and a very important cytokine as well, IL-10. And this interleukin 10 has a negative regulation on both Th1 and Th2 cells, in which when it is bound in the vicinity and activated, so it binds, and then essentially, remember, you're producing IL-2. It has IL-2 on its receptor. It has IL-2 receptor on its surface. So the IL-2 that's being produced by the T helper cells. Let's see if I can get this. IL-2 can activate these cells, and these cells will then over time start, and there are a lot less of them. So for example, if I was to take out of the blood, and when we analyze in the lab the T cells from a particular person, you'll see in that person, you'll see, for example, anywhere from 70 to 80% of the cells are CD4 positive. They're well, 60 to 70% are CD4 positive or so. Another 20 to 25% to 30%, somewhere in that range, 20 to 30% are going to be CD8 cells, and then about anywhere from 1 to 2 to 6% of those cells will be T regulatory cells, and so they're a much smaller number. But if they, in the process, as they start, when an activation in the system is going, IL-2 is being produced, the small amount of T regulatory cells can start to expand, and as they expand, they're signaling out the signal IL-10 to start shutting down the immune response. And so they regulate that and start to tune it down. And that IL-10 basically will now stop the CD4 cells. They start to stop those cells, cause them to potentially go on into cell death, and stop to tune them down so that they are not being activated. And this can happen in which, for example, if a CD4 cell, in this case I've got, a binding to a antigen processing cell, but say this is a self-peptide too. Say this is a self tissue, right? It's a tissue, right? So there could be, say it's the liver or the spleen or something. If this is happening here, in which you're getting activation, as it turns out, there was a T cell, say, that was bound to antigen. There may be an antigen processing cell, a macrophage cell that's in there that's processed and unfortunately has put out a self-peptide, and then it's going on to activate the system in that tumor, and therefore cause potentially an autoimmune disease. Then these T regulatory will also shut that down. And these T regulatory cells will migrate to the periphery and into the tissues to do that. Interestingly enough, tumor cells are able to attract T regulatory cells into them. And this is a major problem for the immune response, because if tumor cells are able to sort of load themselves with T regulatory cells, and they do over time, then they basically shut down any immune response to the cancer. And so the cancer cells that can be then activated, and there's basically two ways that tumor cells try to avoid being recognized by the immune system. One of those is T regulatory cells to shut down the T cells, T cell activation, and the second one is actually tuning down and eliminating the production of MHC1. And so you'll see you'll see actually over time the immune cells, the tumor cells that will be selected for that do not express. The ones that will be able to proliferate the most will be the ones that don't express very much MHC1, if at all. So those are mechanisms in which tumor cells can take on this and try to subvert the system by loading themselves with T regulatories, or eliminating their ability to produce MHC1 cells. These things can potentially be overcome, and that's an important part of understanding modern immunotherapy, and we'll discuss that later. So here are some review questions. So there's 3 review questions. You can stop now and answer these questions. On the next slide will be, of course, the answers, and this lecture will be done. So we are finished for now, and we'll pick up the rest, the secondary organs, in the next lecture.